I'm kind of one of these researchers that sort of came into this issue. I started with studying really originally developmental issues and then moved into the gut and then I guess ended up where I am today. So my lab focuses on what, postprandial response is like, what's the physiology of the gut? How do we digest lipids? How organs interact to create physiology as we know it, and what are the big open questions in the field? Let's see the arrow. Does that work? Yeah, okay. So, I mean, this area from the perspective of human biology is really changing at incredible rates. I mean, fat is bad, dietary cholesterol is bad, dietary cholesterol is good. I'm the generation that had William Shatner on the TV telling us to eat margarine. That turns out to be the worst lipid you could eat because of the hydrogenated fats. Like, if you pick a lipid to cause bad things, it would be hydrogenated fats. And so I really teach everyone in my lab that when we say statements about any health claim, that it's evidence-based. And that is often a challenge right now because understanding how to advise people, what's the problem? Understanding how to advise people on some of these issues is still in flux. Clearly, our society is becoming ginormous. And this is happening all over the world with many people who are obese. And now we have children at record numbers showing signs of metabolic dysfunction. And we know diet plays something to do with it. And the zebrafish is just awesome to really get at these questions because of the optical clarity. So this is live shot. And for those of you who don't know the physiology, you've got the liver, you've got the intestine. You can throw fluorescent lipids in the water and actually look at the enterohepatic flow. And that's one of the big discoveries that my lab started back in 2000. And therefore, you can actually see the gallbladder contract. You can see the bile come out. And then you can, of course, go to other needs like electron microscopy. And we needed a diet that fish would gorge themselves on. And so what we discovered, it took us a while to come up with like a paradigm that we could guarantee that every larvae ate something fatty. And so we ended up using egg yolk because fish fanciers across the world had often used egg powders, and fish love. It's very palatable. It's like a superfood to them. Then we came across another crazy discovery, which was if you put the larvae on a rocker, it rocks about this frequency, and then you warm it, slightly warm, the fish are in like food heaven, and every rock they will take a gulp. So that means you can take the larvae at 6 days out and see every larvae have food in their gut very consistently. Because to do experiments, you need them all to eat, and you need all to eat around the same amount. So my lab's focused a lot on how do you measure how much they eat and all these issues. And it's really easy to see who ate and who didn't, because there's always a few that just say, I'm not eating no matter what, But you can quickly screen them on a light microscope because the optical properties of the gut change. And the optical property of the gut is not changing. It's not cloudy because there's food in there. It's cloudy because every cell of the intestine has intracellular lipid droplets that change the optical properties, and it gives you that feature. So, it's a little soon now after we ate our lunches, but all the fats in a vertebrate that we eat will eventually end up in a cytoplasmic lipid droplet, and very little leaks out the end of a healthy person. Not to be too graphic here, but it's an incredibly efficient system to extract nutrients into those cells. Those are enterocytes. And my lab, I'm going to tell you today about two technologies. In order for you to understand how important diet is to fish, I need to tell you a little bit about what we were trying to do and some tools that are not published even yet, most of them. In all cells that we know, when you feed, whether you're talking about a yeast or you're talking about a human cell, you give them fatty acids and they will do one thing with that fatty acid. It will go to the ER and it will make a lipid droplet, cytoplasmic lipid droplet. And if you take a yeast cell and you knock out the genes to make those lipid droplets, you give them fatty acids, they're dead. So that is an essential component that all cells do to manage the flux of fatty acids. But there's other things that certain cells do, like liver and intestinal cells. They make lipoproteins. And actually, the two have certain similarities. They have phospholipids around them. They can be around the same sizes, sometimes very different, but that's a way to export the particle and feed other tissues. So my lab has invented, kind of made tools to study the intracellular storage and the secretion. And one tool I'll show you is we know a protein that marks a cytoplasmic lipid drop. They're called perilipids. And what we did is these things go up right after a high-fat meal. So we just take our fish, we feed them the egg yolk, and then we fix them, and we can see in purple that perilipin just shot up in the intestinal epithelium. But you don't want to use fish if you're going to just fix them and like make a slide. The whole beauty of fish is to do live studies, to do physiology. And we did that by taking modern molecular tools and inserting a fluorescent protein into the genome of the fish so that we produce that perilipin protein with a green fluorescent tag. And then we got fish like this. So what you're looking at is a live fish under a confocal microscope right after a fatty meal. And if we didn't feed them a meal, it would just be black. So the protein is gone without fat. But within about an hour to 3 hours, these lipid drops get decorated with that fluorescent protein as the fat balls are essentially forming in the cytoplasm. So that's like tool number one. And my lab is kind of exploring these two different plins come onto the drops at different times. And we're trying to understand this. And so this is a frontier question about how is the gut regulating the rate of kind of chewing up these lipid droplets. And we know that these two marker proteins, one in red and one in green, one is plin3, one is plin2, they jump on the drop at different times. So I hope you can see that plin2 comes on at 6 hours, but plin3 comes off at 6 hours. So we're kind of exploring this kind of sequence and how it regulates lipid accumulation in vertebrates, whether it's fish or humans. But very, very little is actually known on in intestinal biology. Most of this work in humans has been done on human cells or adipose or liver. So now that's tool number one. We can see the lipid droplets, okay? And it'll all come together and why I'm telling you all the basic science, because it's all going to be related to the kinds of foods and what happens in fish. So most of you in this audience have had your lipoprotein profile, right? Raise your hand if you've had it. Okay, come on, you know. Okay, so we know there's bad cholesterol and good cholesterol, right? So bad cholesterol is referred to like LDL cholesterol, and this is why clinicians measure your LDL. So all over the world, if you move 20% on that curve, lowering your LDL, your hazard ratio goes down 20%, roughly. So that means you take statins, you go down 20%, you're 20% less likely to have a heart attack. So when I talk to my science colleagues, they're like, oh, should I take a statin, should I not take a statin? I'm like, look, I'm not a cardiologist, but there's a massive amount of data that despite the probabilities of these side effects, this curve seems to hold up. So I always tell people, like, lowering your LDL cholesterol is an important thing. Now, the thing is, what we're actually talking about is there's this protein called ApoB, and the key here is there's one ApoB per lipoprotein, just one. And it's a massive protein, ginormous protein, 500 kilodalton protein. And so it all really boils down these days to, if I could count each of your ApoB levels, I would have a good idea of how at risk you are at this moment of getting cardiovascular disease, which, by the way, kills 25% of the world's population. And even though people take statins and millions of people are living longer, they're still dying of cardiovascular. We didn't cure death. Okay, and so this is still a question of like what we can do. And there are some people who have certain genetics that they will not get cardiovascular disease. So the question is, there's places, what can we do to impact this worldwide killer? Now, why don't people work on lipoprotein biology in fish, in larval fish? Well, that hand is drawn to scale, and that little drop is a fish larvae with its vasculature outlined. So the fish have a nanoliter of plasma. A nanoliter. When they go to the doctors, you usually take a mil. So we need 1,000,000 times more sensitivity to measure lipoproteins. And the way that we did it was putting a light bulb on ApoB. And we put the brightest light emitting protein we could find. It's nanoluke from Promega. It's 140 times brighter than Firefly. And we inserted it into the ApoB gene. And now we can actually see the particles on a gel, and we can see the particles in a fish. So the whole idea that what we're doing is we can study lipoprotein production after eating or in any context by actually imaging the animals. So this slide, and so what can we do with this? Well, we have one of the first high throughput screens now for therapeutics using fish. Using each fish as an experiment in a 96-well plate, we can screen for new drugs that lower ApoB by just measuring the light. Now, here's what it looks like in a normal fish where it's been fasted after development. So after it uses up its yolk storage, it runs out of lipoproteins. And I hope you can see it's getting like light colored here. Then we give them that egg yolk super gorging meal. And you can see it actually takes a while, but within 72 hours, the lipoproteins are just coursing through all of the vasculature of the animal. And then if we kept fasting it at, these are the larval stage, so we start this at 6 days post-fertilization. So it eventually, after 120 hours after in development, you would see it's starting to come down. And so we do this in order to we're setting up this system so that when we create mutations or things that we know cause cardiovascular disease, we can kind of assay it. But we didn't even know what the normal kind of looked like. And just as a proof of concept, if we make a genetic mutation that fails to eat the particles, they get very big, just like in humans. If some of you in this room have this, your particles are big. And similarly, we make a drug that inhibits the production of lipoproteins, we can detect it's very small. So all this is just saying the fish, this little larval fish, we now have created two tools that we can interrogate lipoprotein secretion and storage inside of like the liver of these orchids. This is just to show you that's a wild type at the top. That is an animal that's mutant in making lipoproteins. So you can see the particles are stuck inside the digestive organs. And this guy is unfortunate, carries a mutation that people have in which the lipase that eats the particle can't dock. And what you see is all of the blood vessels are just filled with lipoprotein that's essentially not digestible. So, okay, why am I showing you all of this stuff? Well, we started out getting interested in diets because we wanted to come up with diets that kind of restored the lipoprotein profile. So in this case, we're doing experiments now using gemma, using spirulina, which is a low fat food, using egg yolk, and then we're using diets from the mouse community, the pagan diet, the classic high fat, high cholesterol diets, to try to come up with a model that allows us to say, oh, this is the Western diet. This is like going to McDonald's every day. So it's still a work in progress, but for the first time, we can quickly interrogate how we can do that lipoprotein profile with a larval animal with a nanoliter of plasma. Now, we also interrogate inflammation, and we do that because somebody made a great fish in which every macrophage is labeled in red. And this just shows you, if you put the larvae on a high cholesterol diet, you will see red cells right next to the vein. So this is like considered a marker of what, for some reason, we don't understand why, but the macrophages know there's a lot of lipid and they come to the vein, and that's the vasculature, and that's a part of why people get cardiovascular disease. But what I want to point out is we started to get worried because when you feed this gemma food, this like superfood that many facilities have switched to because you can feed it once a day, you notice that there's a lot of macrophages coming to the vessel just on the gemma. See, we didn't expect that because we were supplementing with cholesterol. We got a lot more to come supplemented, but then we saw that we were getting recruitment just from the food. Similarly, this paper came out by Chen et al. Just showing that basically if you feed high cholesterol diets or high fat diets, their body length is longer and their weight is more. So that's sort of how so So basically, if you're a developmental biologist, and it's almost like an evolutionary selection. Zebrafish community wanted rapid development and growth. So the lipids just shot up in these foods. And the more feeding, it does give you a lot of growth. But then you start seeing disease processes and physiology that may not be desirable because the optimization was for embryo production. And so this is a confocal projection. We're just doing this. Like I mean, we took this shot, and it's one of the few things that the PI, like I was doing this picture. So we took a picture of a liver cell with that marker I told you about that marks the lipid droplets. So this is live. So that's a cool thing you can do with a fish. You can put it under a cover slip in such a way that it's sort of smushed down there, but stays alive for 30 minutes. And you can come in and move the confocal over the liver, and those circles are individual lipid droplets within hepatocytes. And the red are all those macrophages. So I was hoping to see macrophages with lipid inside of them, because those of you who know about cardiovascular disease might know about foam cells where they eat lipid. We never found that. But I just want to show you, this is what happens if you supplement a Gemma diet with cholesterol. But here's Gemma just fed normally, and all the green dots, these are the pigment cells. That liver has a lot of lipid droplets in it just post-feeding. Now, the problem is I don't know what normal is yet, because I don't know what's the reference diet. But I have a feeling that if I caught a wild animal growing out in the Ganges River, I'm not sure it would have as many lipid droplets as some of these foods. So I guess what I'm leading you with is that I am now at a point where, coming from the open questions, like I built these tools to understand the genes and potentially find therapeutics that can help us reduce bad cholesterol. And then one of the big problems that many people get is fatty liver. So we built a tool to see the fat inside cells. But now I'm up against, well, what's normal? You know, what isn't disease? And so now we're coming full circle as a basic scientist. Like, I got to figure this out now. We got to know, should there be lipid drops in a larval liver? And so I think that's where I come in. And I think we, as a community, I want to kind of create a high-fat diet, but I want to create a normal diet. And I do think sense is telling me that we probably want to have a maintenance environment where our fish are not hyper-fed. And then we want to have like a breeder environment because the community is going to demand that. But I think we now have some tools to interrogate what is happening at the basic organ level. But I have no answers, since that I don't yet know what normal is. Thank you very much.
Time for a couple questions.
Continuing on this vein of asking what's normal, I guess how much of these phenotypes are dependent on this idea that they have to gorge on a large meal, as opposed to spacing out these meals appropriately. And do you know anything about, I guess not, but about the wild? What are the meal patternings of wild fish?
Well, I mean, that's why I was mentioning to Steve in the hall, and there was colleagues who've gone out and sampled fish from the Ganges, where they eat larvae, they eat all kinds of stuff. And I'd like to try to take a look at that to get an idea of what the organ fat levels are, what the body fat is like, to give us an idea of a stock that's foraging wildly. The other thing is, in this study, we did not do that kind of super feed. In the later parts, I don't have time to say, we just used a standard larval feeding with and without supplemented cholesterol. So for those, we don't put them on the rocker. The rocker method is a way to just take, it's the first meal, actually. All those studies, when you take a larvae at day 6 that has never eaten, we put them on that rocker for the first meal, so then we can get everyone to eat at the same time, kind of synchronizing. But the later studies, we do as like the standard rearing that everyone else does. So,
even if we had the standard diet, we still may not be able to control how much fish ingests. So isn't the better standard, the better control, fasting? Because when we go to the doctor, we have to fast also before they take a blood sample, right? I don't know.
We are doing all those. If you noticed in my first profile, we do the feed, then we do fast. So we're trying to come up with the lipid assay for a larval fish. But you're absolutely right that the fish, we all know in our fish facilities, when you have just 2 fish in a tank, they're always obese, right? And all our feeding programs, in my opinion, are producing obese fish. And then we have tanks with a lot of fish. They're kind of fighting for food because the feeders are not linearly scaling accurately from 2 fish to 50 fish. And so it's really messed up, basically. Density, and I don't know if you're, Kristen, you're shaking your head. Like, it's really messed up. We have a lot of obese fish in a typical fish facility.
We hopefully have manuscripts out that show that the fat level required under the conditions of our study are around 12%, maybe 10 to 12%. A lot
Of total body.
of 12% of the feed.
Of the feed.
10 to 12% of the feed should be lipid. I think Gemma comes in around 19%. You get trout diets and such that'll be in excess of 25 or something, 20, 25, or even higher than that. But as an anecdote, we made a diet that was in excess of 30% feed to fish just to show how bad it was. They performed well until they got a little bit bigger, and then they went downhill in a hurry. So,
Right.
hard to say.
Thanks, Steve. We'll have more time for discussion at the end of the day.